Okay, and activity 7.2 is modeling DNA fingerprinting. And basically we're going to model exactly the same thing that we did in using the DNA profiling experiment and the gel electrophoresis gel that we set up. So let's model what's happening now in that gel. So basically, for DNA fingerprinting, you start with the DNA fragments. In this modeling exercise, you're going to have four different DNAs that we're going to analyze in this activity. DNA 1 and 2. DNA 1 is pink, DNA 2 is blue. Now, the only difference between DNA 1 and DNA 2, there was a point mutation. One base was changed in DNA 1 to create DNA 2. And basically the question you're asking, how will that affect the DNA fingerprinting profile? How will the DNA fragments, will they migrate the same, or they might migrate differently? Now we compare it DNA 3 to DNA 4. If you look closely at DNA 3, the only thing that difference between those, for example, this is DNA 3. A virus came in and inserted a random piece of DNA into DNA 3 to create DNA 4. Now think about this is happening all the time in the course of human history. Something called retroviruses have always, there are viruses that can randomly insert DNA into our genome. It's estimated that 8 to 10% of our DNA right now is represented by the insertion of random pieces of viral DNA. So that said, let's analyze these DNA fragments using hypothetical gel electrophoresis. And this represents essentially the gel box now that we're going to use in this modeling exercise. In the first activity with our actual gel, we had 8 wells. In this case, we only have 4 wells, represented DNA 1, 2, 3, and 4 up at the top. Okay, closest to the negative electrode, which is the cathode, and the positive anode is down here. So when we load our DNA fragments on this gel, they would migrate in this direction according to their size. The scale over here represents the number of base pairs. Remember, we're working with double-stranded DNA, so we're talking about base pairs. So this is 16 base pairs up here, down to 2 base pairs down here. Larger DNA fragments up here, smaller DNA fragments down here. So, how can we model this? Now, if you look in your lab manual, we've created a restriction enzyme. Okay, now remember, restriction enzymes are enzymes that very specifically cut DNA fragments at precise sites. Now, I often ask my students, what is the source of these restriction enzymes? Where do we find them in nature? Think about a group of organisms where restriction enzymes would be important. And think small. So we now think bacteria or think fungi. Bacteria have hundreds of these restriction enzymes that they produce basically as defense mechanisms to cleave up the DNA of invading bacteria. All right, so they create all sorts of natural restriction enzymes, and we've been isolating these enzymes over the years for molecular biology. So I think we have over hundreds of these restriction enzymes from bacteria to fungi. Well, this is a random restriction enzyme that we created called HSC1 restriction enzyme. Heartline Science Center 1, all right? And it tells you, remember, the recognition site is very specific for each restriction enzyme. And the recognition site where it will cut this DNA fragment is represented here. So wherever you see a double C, double G on the 5' to 3' strand, it will cut through both double strands, making a double-stranded cut called a blunt end, and that's where it will cut. So this is the restriction site you have to remember when you do this exercise. That's the restriction enzyme. So with that in mind, we're going to create fragments by looking at these different DNAs. And this is DNA 1. So basically for this exercise, you would read across the top strand. Remember, this is a 5' to 3' to the top strand. And you would look for that restriction enzyme site, which is a double C, double G. Now right here, whenever I find my first double C, double G, what does a restriction enzyme do? Well, it's exactly what the scissors are going to do to this paper. It's going to cut through both strands, creating a fragment. Okay? Am I done yet? No, I have to look for more. Is there another restriction site along that end, along that fragment? Here's another double C, double G, and I'm going to cut again between right there. Am I finished? No. I'm going to continue to read. Oh, that is actually a G right there. So it's a double C, double G. That would be another restriction site that I would cut. Are there any more? Here's another one, double C, double G, and I would cut through both strands. So from that DNA, I created 5 DNA fragments, okay? Now, hypothetically, I would load all those DNA fragments in a mixture in well number 1, and they're going to separate on the basis of their size. Now, it might be helpful when you do this exercise. What you're going to do is count the number of base pairs. And you can just simply count across the top if you want to know, 1, 2, 3, 4. There's 4 base pairs represented in that fragment, and that would migrate down to essentially the 4 position on the gel. Then you would go along and count up all the number of base pairs in each one of these. I think I have 5. I think I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For that fragment, this fragment has 6. It would migrate down here. Remember the 6 fragments here, and this is going to probably be 1, 2, 3, 4, 5, 6, 11, and I think this is a 12. Okay. So basically, you want to trim off some of the excess paper to put them, arrange them according to their size. Okay. So in this case, from that DNA fragment, from that DNA piece, I generated 5 different fragments. Now, you want to repeat this exercise with each one of these DNAs. This is DNA 2. Again, you would go through there and look for specifically where you find the double C, double G on the 5' to 3' end, and make your cuts like we did for DNA 1. Then arrange the fragments generated from DNA 2 in a lane right next to it and compare. Okay. I went ahead and generated all the fragments from those 4 DNAs, and I arranged them onto the gel box, the hypothetical gel box. And what I did, you might want to do this at home when you do this, because you'll be working with, I'm working with colored DNA, so I know which DNA it is. You might want to specifically, every fragment you generate, put whether it came from DNA 1 or DNA 2, so you don't get confused when you generate your fragments. Also, it's nice to, what I've done is counted up the number of base pairs in every fragment and wrote that onto the fragment, so it's easier to arrange them on your gel box. But after we did the experiment, this is what we ran the gel, and this is how the fragments separated themselves in the gel. Okay, so there's some questions in your lab manual you can answer about this. Using this simple DNA fingerprinting technique, can you differentiate, for example, between DNA 1 and 2? Well, you can say, yes, I can, because we generated 5 fragments for DNA 1, and after a simple mutation in DNA 2, we've only generated 4 fragments. So, and the same thing can be said about DNA 3 and 4. DNA 3, we see 4 fragments here, but DNA 4 has 5 fragments. So you can see the pattern is the pattern or fingerprint is completely different between all 4 DNAs. So on the basis of DNA fingerprinting, you can separate those and say that they are different DNAs, which is true. Now, this works when you're working with very small pieces of DNA with a small number of base pairs. But if you're trying to separate the DNA from, for example, animal cells from your pet cat, for example, you isolate the DNA from your cat, which has billions of base pairs, and you would use a restriction enzyme to generate fragments from that huge DNA sample and try to load all those fragments. You might generate hundreds to thousands of fragments and try to separate all those fragments just on a simple gel. That would be impossible because the fragments would be so close together in size, you would have a complete smear on the gel. So it would be impossible just by simple DNA fingerprinting on the gel to separate them. Well, over the years, we've created a technique that allows you to do that with longer DNAs, and that's called the Southern blot technique. Basically, a Southern blot technique is, it allows you to essentially melt the DNA fragments. You run the gel like I showed you here. Even for complex DNAs, you run the gel. You don't stain it like we're going to do for our gel electrophoresis experiment. You would first melt the DNA. When you say melting the DNA fragments, essentially you would separate each double-stranded fragment. And to model this in this exercise, you would simply take your scissors. When essentially, in the first step of the southern blot is to do this. Essentially, you create a single-stranded fragments from each double-stranded piece, okay? By simply exposing them basically to very high basic sodium hydroxide, very high pHs. The DNA strands will separate them from each other into single strands. And you can represent that, you can create them with your scissors, but be advised that they're still on the gel, so they still occupy the exact same position on the gel, only they're represented by single-stranded molecules. And so if you want to do that, you would essentially use your scissors to create single strands and put them right back in the gel in the same position. Okay? I guess we can cut there. Okay, now we went ahead and created single-stranded fragments from all those double-stranded fragments by using the scissors, and this is what they look like. And they're arranged in the same position on the gel as the original. Okay, so basically what happens now, after you create these single-stranded fragments, you would actually blot them over to a membrane and then stain them. And the only way you're going to see these fragments, we're not going to stain them with any kind of visible dye. We're going to use a single-stranded probes, okay? And this is what the single-stranded probe that we created here. Now, a single-stranded probe is created in the lab, and you can see it only has 4 bases, GGTA, in the 3' to 5' orientation. Now, think in your head, what you know about the DNA sequences and structure, what they bind to, what's a complementary sequence to GGTA? Okay, it would be CCAT, okay? And since this probe is in the 3' to 5' direction, wherever it's going to hybridize with the CCAT on the top strand, which is in the opposite direction. So look at the top strand of all those fragments for a CCAT, and I found one right here. And so that probe will hybridize with that sequence. And best I can, I'm going to put it right there. CCAT. So that fragment is labeled. Now what your task then is to find how many of those fragments, where would the probe hybridize? So you look through all those, you're looking for a CCAT, okay? Well, here would be another one right next to the CCAT there. Sorry. We're going to cut there. And so I guess you can cut there. Okay, so we went ahead and I labeled all the fragments, and this is where the probe is going to hybridize, okay? So at the end of the experiment, you can actually see when we visualize this, the only fragments you're going to be able to see are the ones where the probe hybridized to. So basically at the end of a southern blot, you would only see these fragments, okay? And so basically what you can ask yourself through using a southern blot procedure, can I differentiate between DNA 1 and 2? Can I differentiate between DNA 3 and 4 on the basis of the number of fragments that have been hybridized with the probe? The answer to the first question, you cannot differentiate between DNA 1 and 2 because the probe hybridized with the same size fragment. But overlook it between DNA 3 and 4, yes, using southern blot, I can differentiate between DNA 3 and 4 because 2 fragments were labeled with the probe here and only 1 here. And so that's the advantage of using the southern blot procedure.